Namaste. In this session, I will teach you LCMS working principle, molecular ions identification, and molecular mass calculation. LCMS contains four major components, and these components are: first, high pressure liquid chromatography or HPLC; second, ionization chamber; third, mass analyzer; fourth, mass detector. This is the LCMS flow diagram. The function of HPLC is to separate each components of the sample mixture and send the eluted component to the ionization chamber. Here in ionization chamber, eluted components is gets ionized in gaseous phase. Then ionization chamber send the gaseous phase ions into the mass analyzer. Here in mass analyzer, gas phase ions are separated according to their mass to charge ratio or M by Z ratio. Then mass analyzer transfer the ions in mass detector. Here in mass detector detects the ions and convert them into the electrical signal. These signals are processed to generate a mass spectrum. We are going to discuss the mass spectrum in details in upcoming slide. Now LCMS working principle in details. HPLC or high pressure liquid chromatography. It contains two major processes. First, sample injection. The sample is injected into the HPLC system. Second, separation. Inside the HPLC column, the components of the sample mixture are separated based on their interaction with the stationary phase and mobile phase, and separation is governed by the polarity of the components, polarity of the stationary phase, and polarity of the mobile phase. See how two components are get separated. Now each compound elutes from the column at a different retention time. It enters the ionization chamber one by one. In this chromatogram, first this compound means this peak will enter into the ionization chamber. Then after this analyte or peak will enter into the ionization chamber. Ionization chamber. As each eluted compound exits the HPLC column and enters the ionization chamber, it is converted into gas phase ions. This process is known as ionization. Among the various ionization techniques, electrospray ionization or ESI is the most widely used in pharmaceutical analysis because it is soft ionization technique that produces intact molecular ions with minimal fragmentation. In positive ion mode, ESI commonly produces protonated molecular ions, that is M plus H, positive charge. In negative ions mode, ESI commonly produces deprotonated molecular ions, that is M minus H, negative charge. The gas phase ions are then transferred to the mass analyzer. Mass analyzer. Here gas ions are separated according to mass to charge ratio or M by Z ratio. In the pharmaceutical industry, the triple quadrupole mass analyzer is the most commonly used analyzer. It consists of Q0, which is also called ion guide. It focuses and efficiently transfers ions into the mass analyzer. Q1, which is also called first quadrupole, acts as a mass filter, selecting ions of a specific M by Z precursor ions. Q2, which is called collision cell. Selected precursor ions are collide with an inert gas such as nitrogen or argon, producing fragment ions through collision induced dissociation or CID. Q3, which is a third quadrupole, filters and selects the desired fragments, which is called product ions, before they reach in the detector. See, this is the flow diagram of the triple quadrupole. This is a Q0, which is also called ions guide. This is a Q1, first filter. Q2, this is the collision cell, and this is the Q3. Depending on the analytical method, the mass analyzer can transmit the intact molecular ion directly the detector to determine its molecular mass, or fragment the precursor ion in the collision cell to generate product ions for structural identification and highly selective quantitative analysis. Mass detector. The mass detector detects the ion transmitted from the mass analyzer and converts them into the electrical signal. These signals are processed to generate a mass spectrum which display the mass to charge ratio and relative abundance of each molecular ions or fragment ions. This is the mass spectrum in x-axis, that is M by Z, and in y-axis there is a relative abundance. The resulting data are used for compound identification, structural elucidation, and quantitative analysis. Data analysis steps. These are the key steps required for data analysis. First, find a chromatographic peak retention time. Next, extract the mass spectrum at that RT. Next, locate the measured M by Z. Next, determine whether it's M plus H or M minus H or any other adduct. Calculate the molecular weight. Check isotope patterns, whether it contains chlorine, bromine, or sulfur. Examine MS fragments for confirmation. Compare with standard or expected compounds for final decision. Molecular ions identification and mass calculation. In LCMS, molecules are usually appear as a charged ion. It can be operated in both positive and negative mode. Now, in positive mode or ESI, positive, the common ions are M plus H, M plus Na, M plus potassium, 2M plus H. Now question comes, how molecular weight or molecular mass is calculated. Suppose M by Z is 251.2. It means M plus H is 251.2. Then what will be M or mass? M will be 251.2 minus mass of hydrogen, which is 1.007. In this case, molecular weight or molecular mass will be 250.2 Da. Similarly, mass of other pharmaceuticals or compounds can be calculated in LCMS. In negative mode, the common ions are A minus H, M plus Cl. For example, if M by Z is 249.2, what it means? It means M minus H is 249.2. Then what will be M means molecular weight or molecular mass? Means 249.2 plus mass of hydrogen, and in this case it will be 250.2 Dalton. Similarly, mass of another pharmaceutical or compounds can be calculated in LCMS in negative mode. This is all about this lecture. Any question related to this lecture or Pharma Guru course, you can write at this email or you can contact at this phone number. For online course registration, you can visit to Pharma Guru website. Thank you. Let us see you in the next session.